there, this is FemmeSciComm's third vlog. I post videos weekly on women's health research studies. I still feel like this is a field of science that isn't being talked about as much, and when it is, there's a lot of misinformation floating around. I made FemmeSciComm so women can not only learn more about their reproductive disorders, but also learn how to judge the pieces of information they do see. Every paper, every study, every fact has its own caveats and context. So moving on to a paper I found interesting and thorough. Today's post is on a breast cancer study. October is Breast Cancer Awareness Month, and I wanted to help more people become aware of the current research studies regarding breast cancer. Breast cancer research has come a long way. So I wanted to discuss a recent paper I came across and liked that uses triple-negative breast cancer as its model. This paper was recently published in Scientific Reports, Regulatory Mechanisms Leading to Differential Acyl-CoA Synthetase 4 Expression in Breast Cancer Cells. The first author is Melina A. Dattilo, and senior author is Paula M. Maloberti, both from the Biomedical Research Institute at the University of Buenos Aires. So let's get into it. This study aims to find out what regulates Acyl-CoA synthetase 4, or ACSL4, expression in triple-negative breast cancer. First, a little bit of background. Some of you may already know that one way breast cancer is broken up into subtypes are by its expression of specific receptors. These receptors are estrogen receptor, progesterone receptor, and human epidermal growth factor receptor 2. So what are the statistics of these breast cancer subtypes? The majority of breast cancers are ER and PR positive and HER2 negative. 10% are triple positive, 7% are ER and PR negative and HER2 positive. The type I'll be discussing today is triple-negative breast cancer. 13% of breast cancers are triple-negative. Among the triple-negative patients, 70% of deaths occurred in the 5 years following diagnosis, compared with only 44% of deaths in the other subgroups. This suggests that there's a great need to identify alternative therapeutic targets for TNBC. Previous studies from this research group identified the role of ACSL4 in the aggressiveness of breast cancer. They have already shown that there is high expression of the enzyme in TNBC cell lines compared to ER-alpha expressing cells of lower aggressiveness. And so, in this paper, they're aiming to identify what regulates this enzyme in the TNBC subtype. Through bioinformatic analysis of the DNA sequence surrounding the ACSL4 promoter, the researchers found that the sequence contains a site for estrogen-related receptor alpha, or ERR alpha. With this information, they measured how much ERR alpha protein is present in TNBC cells versus ER alpha expressing breast cancer cells. They found that ERR alpha protein expression is higher in TNBC cells than ER alpha positive cells. Then, through ChIP-qPCR, they observed significantly more binding of ERR alpha at the ACSL4 promoter region that they identified previously in TNBC cells versus ER alpha positive cells. Next, they go on to test if ERR alpha activates or represses transcriptional expression of ACSL4. So they knock down ERR alpha using shRNA. This Western blot just shows the efficiency of their knockdown. The Western blots aren't perfect, probably due to ERR alpha not having a good antibody, but let's trust them on the knockdown. With the knockdown, they test ACSL4 protein levels in TNBC versus ER alpha positive cells. Because TNBC cells express more of this protein, knockdown of ERR alpha decreases ACSL4 protein expression in TNBC and doesn't really do anything in ER alpha positive cells. ERR alpha shRNA knockdown also decreases luciferase activity only in TNBC. This decrease of luciferase activity means ACSL4 transcription is decreased when ERR alpha is knocked down. To additionally test if ERR alpha really regulates production of ACSL4, they treat with an ERR alpha inverse agonist XCT790. This inverse agonist means it binds to ERR alpha and induces a response opposite to an activator. Here we can think of it acting like an inhibitor to stop or affect its usual job. ACSL4 protein levels and luciferase activity are decreased with the treatment of XCT790, which adds to the evidence that ERR alpha is activating ACSL4 production. To test the functional nature of ACSL4 on cell proliferation, they individually and together treat TNBC cells with XCT790 and triaxin C, which is an ACSL4 inhibitor. The combinatorial treatment of XCT790 and triaxin C causes significant inhibition of cell proliferation compared to individual treatment of TNBC cells alone. However, one caveat to this figure is that it's missing a negative control. Where is the minus-minus treatment? I'll talk a little bit more on that a little later. Moving on to their final figure. Because the researchers first saw a negative correlation between ER alpha and ACSL4 expression in TNBC, they wanted to see what happens to ACSL4 when ER alpha is overexpressed. They overexpress ER alpha in both TNBC and ER alpha expressing cells. ACSL4 transcription measured via luciferase is decreased with ER alpha overexpression in both cell lines. Protein and RNA levels of ACSL4 are also decreased with ER alpha overexpression in TNBC cells. In ER alpha positive cells, researchers didn't find binding of ER alpha at the ACSL4 promoter, meaning that ER alpha probably doesn't directly regulate ACSL4 in breast cancer, even if ER alpha is present in the environment, which is why it was done in ER alpha positive cells. However, when they overexpress ER alpha in TNBC cells, they do find ACSL4 transcription measured via luciferase is decreased. Not only that, but ERR alpha transcription measured via luciferase is decreased with the ER alpha overexpression. With this information, researchers also determined ER alpha overexpression decreases ERR alpha protein levels in TNBC cells. So what does this all mean? Remember, triple-negative breast cancer is considered more aggressive and has a poorer prognosis than the other breast cancer subtypes, mainly because there are fewer targeted therapies for triple-negative breast cancer. It's hopeful that there are other studies that use ERR alpha as a possible therapeutic in breast cancer. Its inhibition is currently under study as a new strategy for TNBC treatment. Knowing more behind the mechanism of its downstream effects helps tremendously when developing a new therapy. This study pinpoints one of ERR alpha's downstream targets is ACSL4, which may play a role in inhibiting cell proliferation, leading to the clinically observed high aggressiveness of TNBC. But don't forget the caveats. One, for example, in figure 6I, the negative control is missing. What is the basal level of percent inhibition? Negative controls are important to include in experiments. What if the basal level of percent inhibition is 100%? Then treatment with XCT790 and triaxin C decreases percent inhibition, i.e., increases cell proliferation. Also for figure 6I, I'd like to see the authors expand on this functional aspect a bit. At what point does ERR alpha and/or ACSL4 affect cell proliferation? There are many cell cycle checkpoints and regulator proteins. Does ERR alpha or ACSL4 directly bind to one of these regulator proteins or affect production of one of these regulator proteins? Also, all experiments are done in breast cancer cell lines, and this study doesn't include mouse models or primary tissue samples. A probable next step would be to look at mouse models of TNBC cell proliferation while targeting ACSL4 or ER-alpha. To my knowledge, the use of ACSL4 inhibitors in clinical trials isn't being done yet, but the use of ER-alpha inverse agonists are. So this pathway is a promising therapeutic target in TNBC. Overall, this study adds to the mounting evidence of the importance of precision medicine and individually targeted therapeutic approaches. I hope you enjoyed my video on ACSL4 regulation in TNBC. Remember, it's October, Breast Cancer Awareness Month. Breast cancer research is still advancing and still thriving. Thank you for reading, watching, and listening to my assessment of this paper on breast cancer. If you have any questions, please comment on my video or my posts. Keep informed, stay interested, and keep doing great women's research. Bye. If you're interested in this content, please make sure to subscribe to my YouTube channel. Also, you can check out my website, FemmeSciComm.com, and you can follow me on Facebook, Instagram, Twitter, and Pinterest at FemmeSciComm. Bye.